The cell cycle for somatic cells consists of the interphase and mitosis. Interphase consists of G1 phase, S phase, and G2 phase. Mitosis consists of mitosis and cytokinesis. Interphase consists of about 90% of the cell cycle. G1 phase, which is the first part of it, is when the cell grows. The S phase consists of growing and the copying of chromosomes. G2 phase is when the cell completes preparations for cell division. And mitosis is what we'll describe next, which is when the cell divides. During the G2 phase of interphase, centrosomes have formed, chromosomes have duplicated but are still uncondensed. There's still a nuclear envelope, and there are one or more nucleoli.
The two round objects above the nucleus are the centrosomes. The chromatin is condensing into chromosomes in this phase. Each duplicated chromosome appears as two identical sister chromatids joined at their centromeres and, in some species, all along their arms by cohesions, or sister chromatid cohesion. The mitotic spindle, named for its shape, begins to form. It is composed of the centrosomes and the microtubules that extend from them. The radial arrays of shorter microtubules that extend from the centrosomes are called asters, or stars. The centrosomes move away from each other, propelled partly by the lengthening microtubules between them.
In the prometaphase, the nuclear envelope disintegrates, and microtubules have invaded the nuclear space. These microtubules can attach to kinetochores, or they can interact with opposing microtubules. Additionally, the chromosomes have become even more condensed since prophase, and now have a kinetochore. In metaphase, the chromosomes go to opposite ends of the cell and line up along the metaphase plate. The microtubules attach to the kinetochores. Now things really start heating up. In anaphase, the shortest stage of mitosis, the chromosomes separate after their cohesin proteins are cleaved, and then they start going to opposite poles of the cell towards the asters. During telophase, two daughter nuclei form in the cell. Nuclear envelopes arise from the fragments of the parent cell's nuclear envelope and other portions of the endomembrane system. The nuclei reappear, and the chromosomes become less condensed. Any remaining spindle microtubules are depolymerized. Mitosis, the division of one nucleus into two, genetically identically, then is now complete.
Wow! In cytokinesis, telophase is usually well underway. In animal cells, during cytokinesis, cleavage furrows form before the two cells completely separate. Nuclear envelopes are forming, and the nucleolus is forming. Meiosis is split into two stages, meiosis 1 and meiosis 2. Meiosis occurs in sex cells instead of somatic cells, so it occurs in cells such as sperm and egg cells in humans. The first stage of meiosis 1 is prophase 1. In this stage, DNA coils into chromosomes, the nucleolus and nuclear envelope disappear, the mitotic spindle forms, and synapsis, or crossing over, occurs. Chiasmata is the location in the middle of the chromosomes where the spindles attach in order to line them in the metaphase plate, which we will be talking about next. Metaphase 1 is the next phase in meiosis 1. Tetrads line up randomly along the midline, spindle fibers attached to the centromere of each homologous chromosome. Which they line up is called the metaphase plate, although it is an imaginary line. Anaphase 1 is the next phase. In this phase, breakdown of proteins responsible for sister chromatid cohesion along chromatid arms allows homologs to separate. Homologous chromosomes move to the opposite poles of the cell through the spindles.
The next phase is telophase 1. At each pole during this stage, there is a complete haploid set of chromosomes, but each chromosome still has two sister chromatids. A cleavage furrow appears, and by the end of this stage, the parent cell has divided into two daughter cells. This separation of the cytoplasm is called cytokinesis. In some organisms, nuclear envelopes appear briefly at this point. This intermediate stage is called interkinesis. But in others, the daughter cells begin immediately to prepare for the second meiotic division.
Meiosis 2 is the next stage of meiosis that follows meiosis 1. This is summarized as the separation of sister chromatids. Each cell in meiosis, which is 2 cells in meiosis 2, now, after meiosis 1, each has 23 chromosomes. They will now split and go through the same stages as mitosis, and they will split into 4 total cells, 23 chromosomes each.
Meiosis and mitosis are very similar cell processes, and that's because they have similar subprocesses, such as prophase, metaphase, anaphase, and telophase. Additionally, the two have a similar end result, which is dividing chromosomes. Now, they also follow the same pattern of first duplicating chromosomes, then lining up in the middle to be divided, then being divided and becoming 2 new cells. Additionally, they both use the mitotic spindle, and they also use asters and metaphase plates. However, it's important to understand the differences between these super rad cell moves. The differences, for example, mitosis only occurs in somatic cells, while meiosis only occurs in reproductive cells. Additionally, meiosis produces haploids, while mitosis produces diploids. Also, meiosis produces 4 reproductive cells, while mitosis produces 2 somatic cells.
Hello, this is Preston Thuy here with Urkel Fitzgerald, here to talk about little something about mitosis and meiosis. What do you have to say to us here, Mr. Fitzgerald?
All right. Well, actually, what I'm going to be talking about is interphase. So basically, interphase takes up 90% of the cell cycle. Bet you didn't know that one. And the way that it works is it is divided into 3. It's divided into 3 phases, which is the G1 phase, the S phase, and the G2 phase. This G phase is also known as the gap phases, which is it's like the gap years. They're like angsty teenagers, just like me. And anyways, so first we're going to talk about G1, which is where the cell is growing, and it is getting ready to go to the S phase, which is a very important part of mitosis because the S phase is where cells continue to grow and they reproduce their chromosomes, which is pretty neat, and it's the only reason why mitosis can occur and still result in 2 full sets of chromosomes. So, after the S phase, which we know is pretty interesting phase because they're dividing, we go to the G2 phase, which is where the cell is finishing up, getting ready for mitosis. It's just before mitosis begins, and you got this big ball of chromosome spaghetti. That's what it looks like. It looks like spaghetti. That's how I remember it. It looks like spaghetti, and it's uncondensed chromosomes. However, the nucleolus is still apparent, and that's
Alright, that's all we have here, folks, with Mr. Urkel Fitzgerald.
Why are you cutting me off?
That's great. I'm sorry, sir. Continue.
We're gonna get to commercial. We'll be right back.
Alright, so I was right in saying that we were done. Thank you for watching our video about interphase and the cell cycle and mitosis and meiosis and all their similarities and differences. Hope you enjoyed.